Today, we'll be discussing fungal toxins. Fungal toxins or mycotoxins are low molecular weight natural products produced as candy metabolites by mycotoxigenic fungi like Aspergillus, Alternaria, Claviceps, Fusarium, Penicillium, etc. The term mycotoxins was coined in 1962 in the aftermath of an unusual veterinary crisis near London, England, during which approximately 1 lakh turkey poults died. When this mysterious turkey disease was linked to peanut, means groundnut meal contaminated with candy metabolites from Aspergillus flavus, it sensitized scientists to the possibility that other occult mold metabolites might be deadly. Mycotoxins constitute are toxigenically and chemically heterogeneous assemblies that are grouped together only because the members can cause disease and death in human beings and other vertebrates. Mycotoxins represent a very diverse group. 300 to 400 different mycotoxins have been recognized that differ from each other structurally and in their toxicity. Aflatoxin B1, AFB1, is well-known mycotoxin for its great public health impact. Mycotoxins cause a toxic response known as mycotoxicosis. Nephropathy, various types of cancer, hepatic diseases, various hemorrhagic syndrome, and immune and neurological disorders are the most common diseases that can be related to mycotoxicosis. Mycotoxins are often detected in damp environments and they occur most often in a wide range of moldy foods found all over the world. Mycotoxins are generally produced during the growth or storage of vegetable commodities like cereal grains, nuts, corn, coffee, wine, beer, and grape juice, then sorghum, apple, pulses, and some animal-derived products. The fungi that cause mycosis can be divided into two categories: primary pathogens and opportunistic pathogens. Primary pathogens affect healthy individuals with normal immune system, while opportunistic pathogens produce illness by taking advantage of immunocompromised hosts. The majority of the human mycosis are caused by opportunistic fungi. Next is major mycotoxins. First is aflatoxins. The aflatoxins were isolated and characterized after the death of more than 1 lakh turkey poults was traced to the consumption of mold contaminated peanut meal, as I already told you. Aflatoxins are difurinocumarin derivatives produced by polyketide pathway by many strains of Aspergillus flavus and Aspergillus parasiticus. The four major aflatoxins are B1, B2, G1, and G2. Aflatoxin B1 is most potent natural carcinogen. Other aflatoxins include P1, Q1, B2A, and G2A. It has been experimentally proved that when cows consume aflatoxin contaminated feeds, they metabolically biotransfer aflatoxin B1 into a hydroxylated form called aflatoxin M1. The diseases caused by aflatoxin consumption are called aflatoxicosis. Next is citrinin. Citrinin was first isolated from Penicillium citrinum prior to World War II. More recently, citrinin has also been isolated from Meniscus rubber and Meniscus purpureus. These two are the industrial species used to produce red pigments. Citrinin has been associated with this yellow rice disease in Japan. It has also been implicated as a contributor to ports in nephropathy. So, citrinin act as a nephrotoxin in animals. Wheat, oats, rye, corn, barley, and rice have all been reported to contain citrinin. Citrinin has also been found in naturally fermented sausages. Next is ergot alkaloids. The ergot alkaloids are among the most fascinating of fungal metabolites. They are classified as indole alkaloids and are derived from tetracyclic ergoline ring system. Lysergic acid a structure common to all ergot alkaloids, was first isolated in 1934. Ergot alkaloids are produced as a toxic cocktail of alkaloids in the Sclerotia of species of claviceps, which are common pathogens of various grass species. The human disease acquired by eating cereals infected with ergot sclerotia, usually in the form of bread made from contaminated fluoride, is called ergotism or Saint Antony's fire. Two forms of ergotism are usually recognized. First is gangrenous and second is convulsive. The gangrenous form affects the blood supply to the extremities, while convulsive ergotism affects the central nervous system. Next is uh, fumonisins. Fumonisins were first described and characterized in 1988. The most abundantly produced uh, member of the family is Fumonisin B1. They are thought to be synthesized by condensation of amino acid alanine into an acetate-derived precursor. Fumonisins are produced by various species of Fusarium and Alternaria. There is a probable link with esophageal cancer and fumonisins in humans. Fumonisins has been isolated at high levels in cornmeal and corn grits. Fumonisin B1 can cause transient abdominal pain and diarrhea in humans. Next is Ochratoxins. Ochratoxin A was discovered as a metabolite of Aspergillus. Uh, Species is Ochratius in 1965 and recognized as potent nephrotoxin. Members of Ochratoxin family have been found as metabolite of many different species of Aspergillus. Ochratoxin A has been found in barley, oats, rye, wheat, coffee, bean, and other plant products, with barley having a particularly high likelihood of contamination. There is also concern that ochratoxin may be present in certain wine, especially those from grapes contaminated with Aspergillus, Aspergillus carbonaceus. The kidney is the primary target organ. In addition to being an nephrotoxin, animal studies indicate that ochratoxin A is a liver toxin, an immune suppressant, a potent teratogen, and a carcinogen. Next is Patulin. Patulin is produced by many different molds, but was first isolated as an antimicrobial active principle during 1940s from Penicillium patulum, now Penicillium graecifolium. The same metabolite was also isolated from other species and given the names like clavicin, claviformin, then expansin, then mycotoxin and penicidin. It was tested as both a nose and throat spray for treating the common cold and as an ointment for treating fungal skin infection. During 1950s and 1960s, it became apparent that in addition to its antimicrobial, antiviral activities, Patulin was toxic to both plants and animals. So during the 1960s, patulin was reclassified as mycotoxin. Patulin is commonly found in unfermented apple juice. Next is next is trichothecenes. Trichothecenes are produced by a number of the fungal genera, including Fusarium, Myrothecium, then Phomopsis, Trichoderma, Trichothecium, etc. Fusarium is the major genus implicated in producing the non-macrocyclic trichothecenes. Fusarium, uh, they are commonly found in food and feed as food and feed contaminants. Consumption of These mycotoxins can result in alimentary hemorrhage and vomiting. Fusarium graminearum is regularly found in barley, oat, rye, and wheat. The trichothecene are extremely potent inhibitor of eukaryotic protein synthesis. Next is zearalenone. Zearalenone is a secondary metabolite from Fusarium, a different species of Fusarium. These Fusarium species are regular contaminants of cereal crops worldwide. Moldy grain consumption may lead to hyperestrogenic syndromes, then disrupted conception, abortion, and other reproductive problems in cattle. Now, different methods for controlling mycotoxins. Methods for controlling mycotoxins are largely preventive, and they include good agricultural practices and sufficient drying of crops after harvest. Other approaches include developing post-resistant through plant breeding and through enhancement of antifungal genes by genetic engineering. Then use of biocontrol agents, then targeting regulatory genes in mycotoxin development, and diversion of mycotoxin contaminated communities from the food supply through government screening and regulation programs. Thank you.